So I want to give you some background to motivate the talk today. There's a lot of bacterial resistance, and we know about this. This slide essentially shows you about the methicillin resistant staph aureus. And in many countries, especially India and China, you'll find that the resistance is the order of percentage of isolates with the resistance is in the order of 50 to 60%. And this is a fairly recent study from Lancet, which has compiled these numbers. You can see in North America also the numbers are large, whereas in the UK it's less. One of the problems is that today in many countries you can just walk to a drugstore and ask for antibiotics without a prescription. I don't think you can do that here, right? And the antibiotic usage in low and middle income countries again is startling. Percentage of children less than 5 years with respiratory infections treated with antibiotics, and you'll see, the dark blue, a large portions of the globe have, between 75 to 90%. So you're actually treating kids with antibiotics leading to resistant strains from the early days. So what I'll talk about today are bacterial infections mediated through the pore-forming route. These are called pore-forming toxins. And I'll run you through how pore-forming toxins work, and this will lead into my talk. These are expressed by bacteria, and about 30 to 35% of bacteria are known to mount their infections by releasing these pore-forming proteins. And they essentially work by membrane binding. They work by binding to the membranes. And once they bind to the membrane, they oligomerize to form large pore-like structures. And these pore-like structures essentially insert into the membrane, the mechanism of which is something we'll try to unravel. Of course, they are driven by membrane composition and so on and so forth. And this leads to unregulated pore formation, kills the cells. It can be non-specific or specific. Some toxins require cholesterol and not all. Some of the diseases that have been associated with these are, some of them listed here, pretty virulent ones: anthrax, cholera, a lot of food poisoning that we experience, pneumonia, gastritis, and so on. Also, technologically, there's been nanopores for sequencing and detection. There's Oxford Nanopore, where they've used this nanopore technology for looking at sequencing various other molecules. There are many reasons for bacterial pore formation. One is basically ion imbalance and lysis. So basically, pores form, unregulated pore formation ruptures the cell by leading to ion imbalance. You also have pores which are created to permit the entry of lethal factors. So this is how anthrax works, and then enters the cellular contents, the nuclear contents, and disrupts the cell. You have another means which is again very intriguing. There are some bacteria which, like Listeria monocytogenes, which has this pathway. It enters the cell, it gets endosized into the cell, and it's encapsulated in a vacuole, as shown here. To escape the vacuole and continue replicating, it creates a pore, ruptures the vacuole, and then gets out. So there's a fine balance in pH, which is tuned for this process to work. So what I suggest, if you have any questions or doubts, since we're a small audience, please interrupt, stop me, and ask me any doubts, okay? So that we can have a discussion. So the toxins essentially are expressed, these are monomeric forms, and they assemble into these complexes shown here. The fundamental differences are they're divided into alpha and beta toxins. The alpha toxins largely have alpha helices that penetrate into the membrane, and the beta toxins have beta sheets. They form these beta barrels and penetrate into the membrane. And again, as I mentioned, 30% of infections are PFT mediated, and some examples here. What we've been looking at over the years is looking at kinetics and pathways for assembly and pore formation, largely looking at experiments where we can glean this sort of information out and build models on top of them, correlate pore formation with membrane composition. I'll spend a lot of time on looking at how we've unraveled the role of cholesterol, understand membrane reorganization and repair. This is something new, and I'll illustrate some examples of this. And also, this is something that we started working on. We haven't made much progress. Therapeutic strategies to combat resistant strains, where instead of actually targeting the bacteria, you disrupt the pore formation mechanism. Okay. So some of our ongoing work, I'll just sort of give you a broad view on what we do in this space. We've been looking at the bacterial membrane and trying to understand, build models for the bacterial membranes to understand how actually molecules transport through these membranes. So this is a gram-negative bacterial cell membrane. This is the inner peptidoglycan cell wall. This is the outer membrane which contains lipopolysaccharides, and this is the inner membrane, inner bilayer composition. So we've been trying to look at translocation of antimicrobials, insertion free energies, and with my colleague in physics, trying to look at how antimicrobial resistance evolves and what are the signatures of resistance in the dynamics of the lipids, because he does a lot of FCS with super resolution techniques. Today's talk, I'll focus on pore-forming proteins. So the essentially primary target is the cell membrane. And we'll talk about the complexities and try to unravel a process of pore formation for one particular toxin. So this is something all of you are familiar with. The basic cell membrane structure here, the more complex view that is shown here. And we've looked at the experimental methods that we use, largely done by my collaborators in the experimental groups. We're looking largely at supported bilayers, where we do single molecule, a single particle tracking, super resolution STED microscopy, where we can actually now track lipid and protein dynamics. At a slightly larger scale, we also look at vesicle leakage kinetics. So here you can look at timescales in the order of minutes to see how the pores form and how leakage takes place. I'll talk to you about some of the experiments we've done in that space. More complex system is we are looking at erythrocytes, and this is largely used as an assay by the biologists to see if the proteins that you have made in the lab actually are functional. So you can look at erythrocyte leakage here as an example where you've exposed it to the toxin and you see the color change due to the hemoglobin leaking out. So you can actually monitor the hemoglobin leakage and actually try to understand the kinetics. We've also looked at cell lines, and there's a lot of work, fair amount of work being done on trying to understand repair mechanisms. This I'll again very briefly show you some recent work that we've started in this area. Any questions so far? Okay. So here's a multi-scale view of what we do in the lab. We look at our lab really works in this space, molecular dynamics, where we're looking at atomic scale description, and looking at mesoscale, where we actually group atoms into larger beads and then assemble them. Of course, there are a lot of challenges in looking at mesoscale dynamics, but it can give you a certain kind of information that is useful, and I'll show you some examples of that. And at the continuum level, again, we can model erythrocyte leakage, vesicle leakage to get kinetics on the order of a few seconds to minutes. Now, this connect is important for us because over the last decade or so, we've had access to super resolution methods where with a recent STED that my colleague has in physics, we can go down to 30 nanometers. So we can start looking at lipid dynamics using fluorescence correlation spectroscopy down to scales of 30 nanometers. And that's getting close to the MD simulation scales. Of course, the time scales are way off, but in terms of length scales, we can actually start to sample systems at these scales. So it's a fascinating time. I'm not going to show you so much of a connect with the STED FCS, but I'll show you some of the connect that we have with the single molecule experiments. And for those of you who don't know about MD, essentially it's Newton's second law. The typical time steps, you have particles, you have force fields, and the typical time steps you integrate all atom calculations the order of 2 femtoseconds. If you're coarse grain, you can go up to 10 femtoseconds. And you need a potential V, which is again governed by all the interatomic potentials, electrostatic potentials, hydrogen bonding, and various angles, angle bending, bond stretching to describe the proper properties of the molecules. And again, here a connection with the experiments is you have a low resolution dynamical data on one side, you have high resolution structural data with X-ray, cryo, AFM, NMR, and so on. And you have super resolution, which bridges the two, and we connect that with the MD simulations, which actually gives us atomic scale information about the system we are studying. So in this work, we focus on one particular toxin. So when we started this work about, a decade ago, we actually had got funding for a STED microscope. And then the idea was, what do we study with it? What molecules do we look at? So we rushed over to my colleague in biology and we sort of brainstormed a bit. And she said, let's start looking at pore-forming proteins. And it's been that's the journey that I'm going to sort of walk you through over the rest of the slides. So we looked at this particular protein, cytolysine A, which is expressed by E. coli, and the crystal structure had just been published by Nenad Barnes group at ETH Zurich, a fairly detailed structure. And there were very few structures of the alpha toxins. Most of the structures that have been around are for the beta toxins, because the beta barrels stabilize the entire pore complex in the membrane. So there's been a lot of AFM work done with the beta toxins, and as well as a lot of crystal structure data available. Although now we are seeing more crystal structure coming out with cryo-EM. So this is the monomeric form. So this is how the bacteria expresses it. And once it inserts into the membrane, it oligomerizes into this pore-like structure. But the molecule now, this is dodecameric, but the molecule now in the pore structure takes this conformation, right? And what we see here is that this little motif, which I'll be referring to through my talk, known as the beta tongue, this undergoes a transformation to this helix turn helix motif. And what you find is that the other large conformational change is this helix, alpha A1, flips out and enters the membrane. So you can see this, the blue portion, which is the alpha A1, and this is the helix turn helix motif, which is inside the membrane. So these are these are one of the largest conformational changes that take place in the proteins in this pore-forming family of proteins. More recently, we've been looking at these structural transitions using enhanced sampling methods, and I'll show you some of that data. This structure here, the beta tongue, is largely consists of hydrophobic residues. So it likes to penetrate into the membrane. And then there's this reorganization that takes place. For those of you who are interested, it's about a 303 residue long structure, and quite a large system for us to simulate. So the entire pore complex in the membrane with water, all atom, are typically about 500,000 atoms, in total. And today we can simulate a few hundreds of nanoseconds per day with the computing resources that we have. So there have been some paradigms of membrane oligomerization. Initially, you have the monomer. This binds to the membrane, undergoing a conformational change. And there are these two broad models, growing pore, pre-pore. So I'll explain what they are broadly. In the growing pore model, you have a situation where the monomer actually inserts into the membrane, and then the oligomerization takes place. So once it starts to oligomerize in the membrane, you have partially formed states, which can cause leakage and ion imbalance. In the pre-pore model, which has been shown for other classes of proteins using AFM, you can actually trap these in these pre-pore states. And once they insert, there's a change in height, and you can essentially show the insertion process being taking place. So you can classify these pre-formed non-inserted states and this fully formed inserted state. So finally, the game is that you reach this state where you lyse the cell. Whether you go through the growing pore or pre-pore model is an open question. The other question is, do these undergo a sequential mechanism of oligomerization, a classic chemical kinetics kind of problem, or do they undergo a non-sequential method? And what are the nature of the intermediates? Do they form pre-pores, arcs, and so on? And can we distinguish between the oligomerization pathways? So this is largely the broad questions that we set out to answer in this work. So let me start with some all-atom MD simulations that we initially carried out. So from the crystal structure, you start at initial times, you assemble it as per the crystal structure, and then you run a simulation. Now we can run these for several, up to a few microseconds we've run this. And it's essentially to show you that the structure that you've got is stable and that the force fields that we're using to study the interactions are reasonable. This is a single monomer in the membrane. And you'll find that when you run these simulations, the single monomer, they remain in the membrane. Of course, we've started with the fully formed pore-like state. They remain in the membrane, but there's a lot of motion that's taking place. But the moment you dimerize, you see increased stability. So let me see if you look at the root mean square fluctuation of the C alpha atoms, this is the monomers. You see a lot of flopping around. And if you look at the dimer, you see that the fluctuations have dropped. And this is the full dodecameric pore complex. So you can see the dimer really, as soon as you start to oligomerize, you see the enhanced stability during the oligomerization pathway. Now, yes. So these are all at 37 degrees. Yeah. And these were done with POPC. Because we've done them with DMPC, POPC, with cholesterol. There's not, I mean, these are broadly, similar in terms of the information that we get. There is hydrophobic mismatch and other things, and you probably would be able to pick them up in vesicle leakage experiments and so on. Then we also did some initial free energy calculations with this so-called Martini coarse-grained model. So essentially what we do is we do a pulling simulation of the entire protomeric state of the single molecule, pulling it out and look at the free energy that you have. And you see a very favorable free energy around 50 kilojoules per mole, right? So this really wants to enter the membrane and it's happy to be in the membrane. And as you pull these out, you start to see some disruption of the states. And if you do this in an all atom state, you'll find that you'll start to see these sort of events taking place where there's some disruption of the structure, clearly indicating that it's not happy in the aqueous environment. This is the state in the membrane. So this is a simulation done where we've pulled it out and placed it in the aqueous environment. So this is just to summarize this. I'm starting with a single protomer. We'll build the story up to the oligomeric states. But in the presence of a single protomer, this is an MD snapshot. Ignore the coloring, just focus on the blue, which is the water. We find that you can form a full water channel. And if you look at the structure in more detail, there is a hydrophilic and a hydrophobic phase is amphipathic. And the hydrophilic phase faces the water channel and basically allows water to actually pass through the membrane. So even a single protomer, if it's bound to the membrane, can start to cause leakage. And a lot of cells sense this. And I'll show you towards the end that this sensing actually triggers off a lot of repair mechanisms. I think we have also evolved. Bacteria have been around before us, but we've evolved to actually combat these and get our immune systems to function. So next, we wanted to look at the transition. Now, if you're doing a simulation, or even an experiment for that matter, how do you actually track the transition from a monomer state to the protomer state? That is the state that the bacteria releases the protein to the final state that it's inserted in the pore complex, right? That's a challenge. So we started using these structure-based models or GO models. And these were developed by Jose Onichik and co-workers, largely to look at protein folding. So the idea is that you have either an unfolded state or a folded state, or you have two different folded states. And can you get information of the transition going from one state to the other, right? So we collaborated with Sachi Gosafi, a colleague of mine at NCBS, where we looked at this transition using this GO model. So in a nutshell, what it does is you code in both different states into your potentials. So you have dual potentials, which contains information of the monomeric state that is here, as well as the protomeric state, which is the membrane-bound state. And the barriers that you have in the potentials are all the order of a KT or so. So they're very low barriers. The idea is that if you start with one state, can you transit to the other state, right? So that's in a nutshell how these models work. And they're successful up to a point. And I'll show you the kind of information that we get from this. So this is the monomeric state. We also had to add a membrane in to be consistent with this structure-based model, so that we were able to do. And this is the monomeric state, which is the crystal structure is known. And it's only based on the atoms, C alpha atoms that you have. And again, here is the structure, the membrane inserted state was structure that we know. The idea is, can you go from this state to this state? There's no solvent, right? So it's just the structure that's built in. So hence they're known as structure-based models. So let me sort of show you how this works. So this is a simulation of, so we're going again, the story is going from this state to this state and try to understand how it actually membrane binds and what motifs go in and so on. Recollect that we had this beta tongue motif converts to a helix turn helix motif here. And we have this alpha A1 helix outside, which inserts into the membrane, right? So let's play you this movie here. So we start this. This is the membrane there, and this is the protein. So we can run several hundreds of replicates because these are fast simulations. And you'll start to see that the green part, which is the beta tongue, starts to insert with the membrane, on top. And if you continue the simulation, there. I think the red is the alpha. The colors are switched here. The red motif here, which is the alpha A1 helix, now starts to insert into the membrane. And finally, it will assume this membrane inserted protomeric state. So you can run these simulations several times and start to get information. You can also use these simulations to determine Fret pairs. And because you can start to distinguish between pairs that will give you information about the different states. So there's a lot of information you can actually pull out of these kind of simulations. So what does it tell us? So if we start from the left here, what we run is about 100 replicates were carried out. For 80% of the time, you start with this IM1 intermediate with the beta tongue inserted. Okay. The rest of the time, you find that the alpha A1 helix inserts to give you a second intermediate, which we call the IM2. And there's some transition between IM1 and IM2. Finally, there is, it goes to this membrane parallel version, where the entire protein now sits on the membrane. And this is interesting. I always tell students that when they're making schematics or look at papers with their schematics, that's not how the real world is. You can see a molecule sitting there. A molecule has all degrees of freedom. It can do whatever it wants. It can sample all the available space. So you actually see that happening here. And then finally, you find this peeling off and this forming this protomeric structure. And interestingly, an unadvanced paper, which is just based on crystal structures, there was a part of the paper where they talk about the pathway, and they basically come up with this membrane parallel state as one of the intermediate steps into pore formation. Now, what does this tell us? Essentially, it's telling us that the beta tongue, so if you look at the ratio of insertion times, we find that the beta tongue insertion occurs first, and I'll connect that to some work that we're doing later. Now you can look at some order parameter maps, which will tell you that this state in this two variable space belongs to a protomer and this state belongs to a monomer, just based on contacts. Right? So this is without a membrane, we find that the contacts are largely in the monomeric state. But when you add a membrane in, you'll find that over time completely switches to this protomeric state. So you're able to actually go from one state to the other. So that gave us some idea about how a single molecule might interact. This is coarse-grained again, there's not so much information. The next part of my talk, again, I'll talk to you about how cholesterol plays a role. Okay? And this is an interesting story because for the alpha toxins, there was really not much information on what role cholesterol plays. And I think we've been able to unravel that in a very detailed manner over a bunch of different studies using MD simulations, as well as the single particle tracking that I'll show you in a short while. There's a lot of information on cholesterol-dependent cytolysins, right? So these essentially are known as cholesterol-dependent cytolysins because there are motifs in the protein which bind to cholesterol. So if the membrane doesn't have cholesterol, it won't form a pore or won't bind. But in the case of the alpha toxin, this information was not known, right? So this is an example of a calculation we've done with Listerio, with LLO, listeriolysin. So you find that these are the motifs here. It's the membrane inserted state. And you find that these are all the cholesterol molecules binding to these motifs, right? Now, in the case of the alpha toxins, it's not very clear where cholesterol would bind because there are no distinct cholesterol sites being implicated. So what is the role of cholesterol? So that's what we sought out to answer. So here my colleague Rahul Roy had, came in. He does a lot of turf microscopy. He had just joined us from Sunny Shee's lab. And when we tossed this problem to him, he said, yes, we can maybe start looking at this problem. And the student that we had working sort of jumped onto this because it was all very fascinating for him to track single particles and so on. So we just use supported lipid bilayer. I think all of you are familiar with this. You can use polymer cushions, and then you can start to fluorescently tag lipids or proteins or nanoparticles and do fluorescence correlation spectroscopy and actually pull out a lot of information. So what you do is here you have the supported lipid bilayer on a polymer cushion, and you flow the protein over the membrane. It's in picomolar concentration, right? You want it to be in very dilute concentration, so you can actually resolve the intensities. Let me show you this. This is a movie of what happens. So if you watch this, this is during the experiment, you'll see sort of bright spots forming and disappearing. And this experiment was done with and without cholesterol. Okay, so this is a POPC membrane, initially a bare membrane, and then a POPC cholesterol membrane. But what you'll start to see is you'll start to see the spots slowing down and collecting on the membrane with time. Now, from these, from this image, you image process it, and you can actually get the displacements, right, as a function of time. And from the displacements as a function of time, you can collect a lot of information. So that's essentially what the experiments do. So it's a turf instrument, and there's an evanescent wave, so that you're actually sampling at the at the bilayer interface and tracking the protein. So here, what we did was this is CLI, which has been tagged, and you do the single molecule particle tracking experiment. We also do vesicle leakage experiments to show the role of cholesterol and erythrocyte leakage. I'll just show you this. So here you have calcine, which is a dye which is quenched when it's inside the vesicle. And these vesicles on the order of 100 nanometers, so they're small unilamellar vesicles. And if you look at the intensity here of the leakage, you can actually track the leakage intensity of the dye versus time. What you'll find here is that for DOPC, that is without any cholesterol, the kinetics are the slowest. The moment you start adding cholesterol into the system, you start to see that the kinetics are much faster, right? So cholesterol clearly enhances pore forming activity, and the question is why? So if I show you what we inferred from the single molecule experiments, let's just focus on kind of a busy slide. This is again just showing you that these are the motifs that can interact with the membrane. And these are the case here where you have interactions with the alpha helix. This is the case of no cholesterol. This is the case of cholesterol. So let's start here. This is just a distribution of the instantaneous square displacements. So you're starting this from 100 milliseconds, that is the red curve, and going up to 30 minutes. So you find that the distribution largely remains in the case of no cholesterol. It's much more, it's faster. You're starting to see a peak with slower displacements, but by and large, you're finding that this peak over a period of 30 minutes slowly starts to emerge. In the case of cholesterol, on the other hand, you'll find that this is 100 milliseconds. And in the case of 5 minutes, you move to a slow moving population, right? So cholesterol is clearly having a role, playing a role in actually slowing down the dynamics of the molecule. Now you can ask, are you tracking a single molecule or two molecules? Firstly, you're using low copy numbers, and you do also photo bleaching experiments. So if you photo bleach, then if you have an oligomer, you'll have as many steps as in the oligomeric complex. So they've done a lot of that work for the kinetics. So if you have 2 or 3 mers in a complex and you photo bleach, you'll have 3 steps. So it actually gives you information about how big the oligomers are. So then we started doing MD simulations. And to be honest, when we started it, I didn't really realize that. I didn't think that we'd actually see anything. These are complex systems, but let me run you through what we saw here. So this is the single monomer complex that's in the membrane. And we started to find cholesterol hotspots. So if you look at these residues here, we started to observe that the cholesterol actually binds to some of these residues, which are in the inserted alpha helix. Then in the beta tongue region also, we found that certain residues have cholesterol hotspots. So there was some indication that the beta tongue that is inserted there and the alpha helix that's inserted here have cholesterol binding motifs, and there are these cracked motifs, which are cholesterol recognition and consensus motifs, which are put out there in terms of the sequence of residues which would bind cholesterol. And we find that there's the tyrosine here has a strong cholesterol binding affinity. So this was our initial simulations, which told us that there is some binding sites for cholesterol. Now, what happens with the full pore complex, which is more interesting? So we did the full pore simulation with cholesterol, and lo and behold, we see this. So what we find is that this is a snapshot at the end of the simulation. And what we find is that these molecules are the cholesterol molecules here in orange. We find that the cholesterol likes to wedge between the green, which are the beta tongues. So the cholesterol molecules actually go in between these beta tongues in this large complex and sit in there. And it's like a cement, it sort of glues the entire structure together. Then you can, since you have MD simulation data, you can analyze this in detail. So these are the beta tongues that you're seeing here, which is in green here. And this is a cholesterol molecule sitting in there. So it lives there for a very long time. So we've run these for about a microsecond. And the residence time of the cholesterol molecule in there is several hundreds of nanoseconds. And if you pull it out, it goes back in, indicating that this is indeed a very favorable site. And there are a whole bunch of residues here which, like cholesterol, especially the Y27. And if you look at the cholesterol density maps again, you see hotspots for cholesterol because of the location shown above. So clearly, cholesterol seems to be playing two roles here. One is it's initially helping the molecule bind. And once the pore complex is formed, it's actually gluing the entire complex together. So it essentially stabilizes the full pore complex. Now, let me walk you through a story which we started to ask, what is the role of cholesterol? Molecularly, what is the role of cholesterol? Clearly, it is enhancing the binding, and it seems to be stabilizing the entire pore complex, at least from both the experiments and the simulations. But we realized that there is a conformational change taking place from one state to the other. How is it helping this conformation? So the holy grail would be actually to go from this state to this state. And in an MD simulation, it's almost impossible to actually solve this large problem that we have. So we focused on just the beta tongue motif. Now, this is actually the string method work, and this is about 3-4 years of work in one slide. So this is done with my collaborators Sudeep and our student Avijit, who spent a lot of time developing these methods and trying to apply them to membrane proteins. So a lot of this work has been done for proteins, in bulk, but we have not seen any studies of membrane proteins. So this is an example where we started to look at membrane proteins. So the idea is the following, that if I start from this structure, which has those two green beta sheets, as shown here, because we first stick it in the membrane, we have not looked at the entire protein, we only looked at a part of the protein. So you can question how realistic this is, but it's a start. And let us see the kind of information we get from this. So we need to make sure that this motif is actually stable in the membrane. So we actually stick it in the membrane, run these for several hundreds of nanoseconds, at least run these for microseconds, with several repeats, and you essentially end up indicating that it's stable. Now, when you have cholesterol in the membrane, what you start to see is that, a spontaneous unfolding. It's going to sort of unfold and refold, right? It's going to refold into that little motif on top, which is a helix turn, helix motif. So we see in the case of, when you have cholesterol, you see a spontaneous unfolding occurring in the membrane. And the final state is this helix turn, helix motif, which again remains stable in the membrane, although we're looking at this single motif. Now, without cholesterol, we have done a detailed analysis of the secondary structure and so on. Without cholesterol, we find that there is very little limited loss of secondary structure. So somehow cholesterol seems to trigger this loss of secondary structure in the membrane. And then we do the string method with path collective variables. These are two, the collective variable space involves two coordinates Z and S here, which involves information from the entire path and hence known as path collective variables. And this is the free energy landscape that you see. And if you plot this as a function of, in a 1D space, this is the free energy as a function of going from one end to the other end. This is the kind of information that you get. Without cholesterol, you see this large barrier in the for the conformation. You're going from, again, this state or this end to that state at that end. The initial states are different with and without cholesterol, but it gives you a very clear idea of the barriers that you're seeing. So this is the initial state without cholesterol. And finally, you get to this state through a large barrier of about 140 kilojoules per mole. When you have cholesterol, there's already an initial unfolding, right? And then there's a smaller barrier to overcome to go to the final state here. Again, here, when you don't have cholesterol, this unfolded state is an intermediate which occurs somewhere here, right? So cholesterol seems to catalyze this whole process. We've got a lot more information on more detailed maps of interaction with various residues and so on, which gives us more molecular insights. So this, I think, gives us the role of cholesterol and tells us how, at least for one part of the motif, how cholesterol is playing a role. So I'll continue from any questions here. At this, how much time do I have, Bart? Yeah.
So why we don't have a value at the reactants?
I mean, we have one over here, right? And this is very weak here in this case. So it's not like a, and here you do have it, right? I mean, if you look at this case here, this is the one end, which is that state there. So this is a nice minima here.
Other side is.
This one here.
Yeah.
So this is weaker because this is the case, I think, with cholesterol. This is the case with cholesterol. In the case without cholesterol, what we find is that this state that you have here, without cholesterol, you find that it's it remains in this folded state. So the various, I mean, both states are very different with and without cholesterol. But these we run vanilla MD simulations long enough to be able to capture the two states. So we're assuming that it's some local minimum. And once we have the data from that state, then we interpolate to the other end state and we create points on the string. You can always question, is this state really the lowest, but it's dependent on how long you've run it. But by and large, it seems okay. Right, yeah. So, yeah, I didn't know how much time do I have? Yeah, that should be good. Yeah. So I'll just run you through this part of the talk, which where we sort of moved on to a longer length and time scales using dye leakage experiments, using small unilamellar vesicles. So what I've shown you up to now is largely single molecule kind of data, showing you how things work at the molecular scale. What happens when you look at kinetics at a slightly larger scale? Clearly, we can't use the techniques that we've used. We have to use other techniques and other methods. So mostly kinetics-driven form. So the idea was the following, that you're looking from a monomer state to a protomer state, and then forming pores. This is the kind of information that I showed you earlier, looking at the different possible kinetic mechanisms and so on. So let me show you this again from the dye leakage experiments. So you can run these experiments with small unilamellar vesicles. These are 100 nanometers. So the curvature is large because the pore itself is in the order of 10 nanometers. So you have a very large curved surface. And, there are barriers to forming pores in these highly curved surfaces. But anyway, nevertheless, we look at the leakage data, and it's in the order of time scales in the order of minutes, right? So you plot the normalized intensity, which you get from the experiment, and you can fit these to two time constants, one which is less than a minute and one which is in the order of a few minutes. So that's the typical time it takes for the pores to form. And the question is, can you relate the leakage to the kinetics? That was the question. So a simple calculation will tell you that you can pull out a time constant just from this simple first order reaction. You can pull out a time constant, which is from which you can actually relate it to the length of the pore, the radius of the vesicle, the radius of the pore, the number of pores, and the diffusion coefficient. So you can pull out this information, and for a single with NP equal to 1, the leakage times are on this scale. This is 10 to minus 3 seconds. For slightly larger vesicles, it's an order of 1 second. And in the case of GUVs, you can go from, depending on the size of the GUV, because it goes as cubed, going from anywhere from 5 to 10 to the power 3, right? Now, if you look at this SUVs here, the leakage times are very, very fast compared to the kinetics of pore formation, right? So if you study the system, what you'll be tracking, if you look at SUVs, you'll be actually tracking the pore formation kinetics and not the leakage. So leakage is instantaneous. What's determining the whole process is the pore formation time. This is what we use to actually glean out the pore formation kinetics, right? So again, we write kinetic equations down. So I'll run through this quickly. We do a monomer balance, conversion of the monomer to, the disappearing of the monomer from the bulk by binding. And that there's a conformational change that takes place monomer to protomer. So the M converts to a P, and then you write down equations, kinetic equations of P. I'm sorry, this is sort of some condensed form. But essentially, it's a balance on all the monomers. And you can write these for different kinetics, that is for sequential or non-sequential and so on. And we couple this with a Poisson kind of process for pore formation. So the leakage kinetics comes from this Poisson process, and the timescales for the actual formation of the different pores are from these equations. So you solve the whole thing. So let me show you what we get. So if you have a non-sequential reversible mechanism, this is the efflux function we get from the leakage experiments. And the non-sequential reversible, you get, you're not able to capture the entire dynamics correctly. And you're always left with a population of single MERS. And this is the population of the MER complexes that you get. And if it's non-sequential irreversible, again, you do not really capture the leakage kinetics. And if it's irreversible, you sort of move towards a higher population of the larger intermediates, because you're depleting the smaller intermediates. So clearly this, at least from whatever data we have and the analysis that we have, the non-sequential mechanism doesn't seem to work so well. But the sequential reversible gives us a very good fit, provided we take, so in the model, we can say leakage is happening due to 5 MERS to 12 MERS, or we can, sort of move the MERS that contribute to leakage around. Right? If you do that, and if you look at this 5 to 12 MER fit that we have, we're able to actually get the experimental data spot on. Very good match with the data. Of course, there's some fitting happening here, and we have constants in the model. So there's a forward reaction constant tau f, which is 4 seconds, and then there is a conformational time scale again, which is on the order of a second, which seems to match with experiments. But there's a large backward time constant of about 170 seconds, indicating that there is some backward, there's some reversibility taking place, but that time constant is small. And with this sort of kinetics, we always pick up some population of a single MER on the surface. And my colleague now, with his single particle tracking with photo bleaching, has been able to show that you always will have single MERS population on the membrane, which seems to indicate this reversible mechanism is at play. And if it's irreversible, again, we don't capture the dynamics correctly. So we think that the mechanism that's taking place is largely the sequential reversible mechanism. So the story that evolved from this was the following, that these assemble on the membrane in these partially formed states, and then they can insert to form the pores. This is the insertion, which causes leakage, which is very, very rapid, on the order of 10 to minus 3 seconds. And the kinetics we're tracking is this process of assembly. So you have these sort of partially formed states that form and insert to form these arc-like states. So the arcs are partially formed pores, which we claim are stable intermediates. And we started doing some MD simulations on this at that time, looking at these very partially formed states. So we set these up from the crystal structure and run these simulations. And what we find is that, let me sort of run through this with you. This is a 6 MER. This we ran for very short simulation times. We've run these longer now. But we find that these are all stable states. So these partially formed arc-like states, these are top views. There's water in here. So the white parts are where we've removed the water for better clarity. So you can find that a lot of these arc-like states are stable. Our MD simulations indicate they're stable. And we also looked in the literature. I'll skip all this. We looked in the literature for evidence of this, and we looked at some old micrographs, 2003, 2014, and we find that indeed you see these partially formed states on the surface of the membrane. So clearly, these partially formed oligomers are present, and they are viable intermediates. You can shift the balance, right? If you have very low population of MERS protein, then you will have a larger population of arc-like states. But if you have a higher population of proteins, you can push it to fully formed pores. So again, it really depends where you're working. The issue with the vesicles is that they are highly curved, and they do not really form the full pores. So they're mostly dominated by these partially formed pores. So these are the kind of oligomerization pathways that have come out. I mean, we are not able to conclude whether it's this or this clearly, but we're able to say that indeed, these pathways are possible. These are stable intermediates. You can form these in the membrane. Our modeling with the lysis kinetics indicates that these partially formed states can form early on and then insert into the membrane to give you these states. So it's some hybrid mechanism really which is going on between these two mechanisms. And then we've said something about the pore forming kinetics. Okay, I'm almost done. So the last part I want to talk about a little bit is about the repair mechanisms and what happens to the lipids, right? So when these pores actually insert into the membrane, where do the lipids go, right? This is a question that one can ask. Now, in the partially formed arcs that I showed you, the lipids actually diffuse out very rapidly. So we've looked at the survival probabilities of the lipids from the MD simulations, and they move out very quickly to form this toroid complex. These are some experiments that we're doing with another class of toxins known as the bipartite toxins, where you need two different kinds of monomers. So they're more complex. You don't have a single type of monomer. You have this AB toxins. Have you worked on any of this? No. So these are the AB toxins. So Yersinia AB. So we've started working on this. We've done some MD simulations, and my colleague Sandhya is doing some experiments on these systems. And let me sort of run you through this. So this is a case, I'll just set it up here. This is some cells, HeLa cells with no toxin. This is with this YXA. This is YXB added after A. So there's a sequence to which is required for you to see activity. I think if you add B before A, you don't see activity. This is you add A and then B, you see activity. So let me run you through this. So this is no toxin. So the movie is playing, you don't see anything happening. And if you look at the case where you just add A, you see some changes, but nothing significant. If you add B after adding A, after 40 minutes, you start to see these blebs being formed. So there's a lot of blebbing from the cells in response to the pore formation. Okay. Now, if you look at 60 minutes, and you can focus on some of these things where the red arrows are there, you can see the bleb forming and disappearing. And some of the blebs will just grow and puncture. So you can again see a bleb form and disappear. These are just bright field images. So now they're doing actually detailed turf microscopy and all this. So it's not clear to us exactly what is happening here. There's more detailed labeling done of the proteins, lipids, to actually indicate what these vesicles are made of. Are there proteins in the vesicles? But there's evidence to show from other studies that a lot of these vesicles bud out to get rid of the toxin and to repair the cell. So what is the fate of lipids during pore formation? That is the question that we ask. And these are, again, some experiments done with my colleague Joydeep and our student Ila, who recently graduated. So again, these are done with listerolysin, which is a cholesterol-dependent cytolysine. And these form pores with different sizes. So these are, again, fluorescence images. And you vary the concentration in nanomolar of the protein and look at the lipid intensity versus the protein intensity. Okay. So the lipid intensity is red, as you see here, and the protein intensity is the green. So you find the lipid intensity is dropping, indicating there's some loss of lipid, and the protein intensity is growing, right? So the question you can ask is then, is there what is the evidence for these as to where the lipids are moving or migrating or being reorganized? What is the source of this? So this is some work done by Vogele and Gerard Hammer's group. I mean, this is a PNAS paper in 2019. And let me play that for you here. So this is a large Martini simulation where they see that once the pore forms, you find this sort of vesicle forming inside. The lipids form this vesicle and it actually moves out. Okay. And we had done a similar work with cytolysine A, actually about 5 years before this was published. And my student had it, when he did all these artwork, he finally did this with the Martini and he said we should publish it. And I said we shouldn't because there's no experimental evidence for it. But it turns out that after this paper came out, we actually sent this to PNAS as a letter. And within a month, I think it got accepted. There was a response from Gerhard Hammer. We saw similar things, with Clive, and indicating that this sort of, in case there is a pre-pore mechanism for the entire pore to pass in, you start to see these, the formation of the micellar plugs into the membrane, and then it starts to eject these out. So it's possible that some of the lipid intensity loss that we're seeing is due to this mechanism. Okay, good. So I'm almost done. To summarize, we've looked at, pore forming toxins, their pathways, spoken about intermediates, spoken about free energy insertion transitions, which was something that we worked on independently, the role of cholesterol, and looking at membrane reorganization events. Finally, let me thank a large number of students over the years who've been working on this, large number of my faculty collaborators. Rahul is the one who did the single molecule experiments, and Sandhya and Prabal are my collaborators. We started working on this problem right in the early days. Rahul is the one who did the single particle tracking. The students here, there's a large number of them. Avijit did all the free energy calculations. Harsh is working on the AV toxins, and so is Samlesh. Pradeep did, Ayush, Rajat, Vaidyanathan, they're all the ones who actually started working on the MD simulations and the single particle tracking. Pradeep is the one who did all the single molecule experiments. And funding sources from the Department of Science and Technology, some funding from Unilever for parts of the work. And we have also a unit of excellence in computational material science funded by the government, where a lot of these calculations are done. And thank you for your attention. A lot of this is published in an Accounts in Chemical Research, which came out in 2021. With that, open to questions.
Questions?